for your informative discussion and your research.
Thank you very
really
much,
appreciate it. Thank you very much.
Thank you. Bye-bye.
So, bye, Sergio. Thank you. So for our next speaker, Dr. Lucía García from Spain, and her topic is on harnessing Drosophila for drug screening.
Can you hear me?
Yes, we can hear you.
Okay, thank you. So, hello everybody, I am Lucía from the lab of Maria Dominguez in Alicante, Spain. And first of all, I would like to thank the organizers, especially Isabel, for giving me the opportunity to giving a talk in this wonderful workshop. I am a junior postdoc, so I am very happy to having this opportunity, and I think I will learn a lot also. Today I will speak about high throughput drug screenings in Drosophila and their potential for biomedical research. So let's start defining what is unbiased high throughput drug screening. This technique basically consists on the screening of large libraries of chemicals for a particular biological activity in high throughput assays. So it can be performed in different disease models such as zebrafish or C. elegans or yeast, and also our Drosophila friends. And of all the compounds that we are testing, we have the aim of finding those candidate ones that are more effective and less toxic. That means that are the ones that work better for our biological activity, and also the ones that have the less possible side effects. Once we have our candidate compounds, it follows the validation phase in other models, such as cell cultures or mice. And finally, if succeed, it would enter clinical trials. These steps involve the management of some complex concepts more related with pharmaceutical companies and economical investments that we are not going to talk about today. But I recommend you to read this review from Professor Roskagan, which is very helpful and explains some of these issues very well. But why do we use flies? As Isabel has already explained in the introduction of the workshop, there are some general advantages of using this model. For example, it has a short life cycle. The housing is easy and cheap. Also, we have an advanced genetic toolkit available. It's also very interesting, the fact that almost 60% of human genes are associated with disease have fly homologs. And also, particularly in the case of studying cancer, it's a good model because there is a similarity between epithelial tissues and susceptibility of tumorigenesis, similar than humans. Also, it is possible to recapitulate the interaction among multiple cell types, the microenvironment, and systemic inputs such as hormones or metabolites in general. And also, it's one of the most important advantages that sometimes we forget to say is that with Drosophila, we can bypass some of the ethical issues that surround the use of vertebrate models. And specifically for drug screenings, the use of Drosophila allows us to address mechanisms of drug response or drug resistance. Also, we can use a large number of animals, which increases the statistical significance and also the robustness of the technique. Also, the drug delivery usually is by feeding, so we can select those bioavailable compounds that are assimilated in the gut. Also, it allows to identify unsuspected cooperative interactions in disease initiation. Also, we can find unknown targets of that are underlying the disease, or different mechanisms that were unknown previously. And also the fact that we do the drug screening in the whole animal allows us to see the interaction of drugs with the emerging tumor cells, the microenvironment, the immune cells, and the organs that support energy adaptation. We work with a Drosophila cancer model that involves the simultaneous hyperactivation of the Notch and AKT pathways, which, when they are together, they cooperate to promote tumorigenesis. Well, what is this? These pathways are highly conserved in humans, and mutations in these pathways are frequently found in some types of human cancer, such as pediatric T-cell acute lymphoblastic leukemia, that from now on we will say TALL. And in these cases, the mutation in these two pathways, it's associated with aggressiveness, resistant to the treatment, with a lot of side effects and gastrointestinal toxicity, and high lethality in relapse disease cases. So our aim was to find compound candidates that can target the Notch AKT-driven cancer with the aim to potentially use this in human cancers. We induce the hyperactivation of Notch and AKT pathways simultaneously in the eye imaginal disc of Drosophila using the ILS promoter. And this, as you can see here, this is a wild-type imaginal disc, and when we hyperactivate both Notch and AKT pathways, we create a tumor mass that in the 70% of the cases, it will be lethal, so the animal will not arrive to the adult phase. And in the 30% of the cases, these adults emerge with full-blown tumors. So this allows us to screen with which compounds can suppress the tumoral phenotype just by visualizing the eyes of the adult flies. Here you can see how it looks like an imaginal disc, a wild-type imaginal disc, and the wild-type eye of Drosophila. And here you can see eye imaginal disc that is tumoral, and the resulting, the emerging fly with tumors in the eye. So let's see with a little bit more detail how a drug screening is performed. First, we do crosses in mass to obtain Notch AKT larvae. And after egg laying, we select those ones that are the Notch AKT positive larvae, that are GFP negative ones. We select L2 larvae. And then we put 15 of these larvae in a tube with the compound that we want to test, mix it in the food. And we do the same with the vehicle of the compound. We do this by duplicate, and also we repeat this for each drug of the screening, for each drug that we want to test. And then we score for the adults. In the adults, we score for the lethality and the tumor phenotype in the emerging flies. Okay, so after scoring approximately 100 and 1,000 flies, we select those drug candidates that suppress tumorigenesis, as you can see here, but also that has the less side effects, because these ones are the ones that we are going to think that can have a potential use in human TALL. I will give you a brief overview of the results of the drug screening, because I prefer to focus in the technique itself. So After 3 double-blind rounds, we found a group of compounds that act as potent suppressors. And among these, we found some anti-inflammatory drugs. The top compound of the screening, it's a lipoxygenase inhibitor called BWB70C, which reduces tumorigenesis at the lowest dose and also causing the less high side effects. Also, it's important to notice that our screening also identified 15 of the 21 FDA-approved anti-cancer drugs that we use as internal controls and that are already established that are anti-cancer. So this highlights the robustness of the drug screening. Because we know that most compounds have a known human molecular target, we validated these results genetically by using RNAi lines of each candidate target genes and seeing if they mimic the action of the corresponding compounds. We found that 64% of the compound act through conserved targets. And this indicates that despite the evolutionary distance between Drosophila and humans, we can still use our Drosophila-based strategy to identify anti-cancer drugs as well as the clinically relevant targets. As I said before, our screen revealed the presence of numerous anti-inflammatory agents that are suppressors of tumorigenesis. And our top compound, the BWB70C, is described as a 5-lipoxygenase inhibitor. So we investigated the contribution of the lipoxygenase-mediated production of pro-inflammatory lipids, and we corroborated that the genetic silencing of the steps of this pathway, of this inflammatory pathway, also suppressed tumorigenesis, confirming that LOX-derived inflammatory lipids are required for Notch-AKT oncogenesis. Our screen also found suppressor drugs targeting the nitric oxide signaling pathway. So we investigated how nitric oxide might be contributing to Notch-AKT induced tumorigenesis. So we observed that nitric oxide synthase, which is the enzyme that synthesizes nitric oxide, that is a molecule that is widely implicated in inflammation, was aberrantly expressed in the tumor imaginal disc. And moreover, silencing this nitric oxide synthase using a selective inhibitor or also by genetic inhibition significantly suppressed tumor growth. Furthermore, we found that aberrant NOS expression was sufficient to dampen the immune response of Drosophila by reducing the melanization innate immune response. So we see a reduction in the expression of PPO enzymes, which are the enzymes that carry out this melanization immune process, and showing a strong reduction of melanized crystal cells. Altogether, these observations indicate that activated AKT pathway unleashes the oncogenic potential of Notch by inducing aberrant NOS expression, which in turn triggers an immunosuppressive environment that facilitates tumor immune avoidance, something that is quite common in cancer. So the drug screening not only allows us to find potential therapeutic compounds, but also serves us to find unsuspected and unknown molecular mechanisms involved in the tumorigenic process itself. Finally, we validated the anti-tumor effect of our top compound in well-established human TALL models that have Notch and AKT mutations. And we observed that the treatment killed all the leukemic cells, which were resistant to Notch inhibitors. That is the therapy that is currently being used in medicine, that had little or no toxicity against normal T lymphocytes. Moreover, similar to the results obtained in Drosophila tumors, we found that endothelial Notch gene was aberrantly enriched in these TAL cells. And we found that our drug selectively killed these leukemic cells by suppression of aberrant eNOS, therefore pointing this compound as a potential treatment for human TALL leukemia. So as we have seen, the use of Drosophila for pharmacological in vivo screenings has a lot of advantages, but there are also some limitations that we must consider before planning to do a high throughput screening. And these are related to limitations in the technology itself. So there is a need for appropriate hardware and softwares to accelerate the process, because currently and usually what we do is that we observe the phenotypes visually, we count manually the phenotype. So this ends up being a very slow and laborious process. So we must take that into consideration, and we should also focus our efforts in overcome this limitation. But also it's necessary to know which fraction of the results that we obtain from a drug screening in Drosophila can be actually translated to human models. So in this sense, the ongoing work with Drosophila avatars is yielding some insights into the translational value of Drosophila screenings. So Drosophila avatars were first proposed by Professor Ross Kagan, and this consists basically in using a Drosophila cancer model that have a combination of different mutations that are frequently found in a tumor from a patient. And then you do a screening in these animals to identify drugs that will later be tested in cancer patients. So this is actually being tested in the Center of Personalized Cancer Therapeutics, where they take the genomics data of the patient to build a Drosophila avatar with the same mutations, and then they screen for FDA-approved drugs. And the results are the results with considerations of toxicity and the dosage and everything are incorporated into a report that will be then given to the oncologist to consider these recommendations. Sorry. Also, Dr. Nahuel Villegas, which is also a part of this Drosophila workshop. It's one of the collaborators. He did the drug screening in our lab, and he is now the chief scientific of a company in UK that is also doing these Drosophila avatars with the same purpose. So although I am not working actually with fly avatars, I wanted to mention these two scientists because I think this is a really exciting step towards personalized medicine, and is particularly important and useful for those rare cancers or those ones that now don't have a lot of therapeutic options. And I admire them a lot, so I wanted to mention them. And of course, I wanted to thank Maria Dominguez for giving me the opportunity of working in her lab and learning a lot, and you for attending, and I will be very happy to answer any question in case you have it. So thank you very much.
Thank you so much, Dr. Lucía. So we have a first question from Helen, I think. you see this. Sorry, Lola. How do these compounds affect normal notch signaling?
How these compounds affect normal notch signaling?
Yeah.
Okay, so these compounds are not targeting notch signaling itself. They are targeting more downstream targets of the notch and AKT cooperation. So actually, we did some trials with inhibitors of notch pathway, and the inhibitors of notch pathway normally have a lot of side effects that we can see in the fly, but also that we can see in humans. As I told you, this gastrointestinal toxicity is due to the fact that we are inhibiting notch pathway that also has a normal physiological role in normal cells. So that's why with the screening, we wanted to target something that is downstream the cooperation of notch and AKT, so we don't affect these pathways because they are important for other normal functions in the organism.
Thank you. We have another question from Helen. And Helen, do you mind if you unmute yourself to elaborate on your question, please?
Okay, thank you. This question is not for this last speaker specifically, or indeed for the last two speakers. We talk often about species differences when we deal with mammalian systems. But each time I listen to Drosophilists present their work, they're always emphasizing the similarities between Drosophila and humans. So I'm wondering, is it that these species differences don't occur between Drosophila, for instance, and human beings? Or is it that we are not looking as at yet?
Sorry, I cannot hear I cannot hear well the question.
Okay, I'll repeat myself and I will go slower.
No, it's the volume that I don't listen very well.
Okay, I will shout. Can you hear me now?
Yeah.
Okay, the question is, have you encountered in your work any species differences between flies and humans when you do these drug screenings?
If we have found differences between flies and humans?
Yes.
Yeah, actually, I mean, as I was telling at the beginning of the talk, there is a similarity of 60% more or less of the genes between flies and humans. And actually, we can see these differences, for example, when we use the FDA already approved drugs in the screening as a control, we can see that in some cases, these FDA drugs are known as anti-cancer drugs in humans, but then in Drosophila, sometimes we see that they are acting as anti-cancer, not all of them. Most of them are also anti-cancer drugs, but some of them do another effect. So that is actually highlighting the fact that even if there are similarities between the two species, of course, there are differences in the metabolism and the pharmacokinetics and other, I mean, there are some differences. So we should take this into account also, that not always everything that we are going to find in Drosophila is going to be totally full translated into humans, but for a great percentage, it does. So it is still worth it.
Definitely. Thank you.
Thank you.
Hamad, do you mind if you unmute yourself, please, to ask your question?
Okay, yeah, no problem at all. Right, thank you so much for the talk. That was really fascinating and really brilliant. So you've done very well. So my question is, so how many compounds were you able to screen in your platform? And also, did you try like probably drug combination? And in your system, did you see metastasis? Like, is it just tumorigenesis or did you see any form of metastasis?
Okay, so there are 3 questions. So how many compounds we screened the LOPAC library of compounds that are 1,280 compounds, something like that. so more than 1,000 compounds. The second question was, sorry?
Was drug combination. So did you try?
So yeah, for drug combinations, we haven't checked. Probably it will be a good idea, because I know that that in humans and also in Drosophila, cancer is a very complex disease. So sometimes the combination of different drugs actually they act synergistically, so it sometimes work better than single drugs. So this definitely would be a very good approach. And finally, about metastases, the answer to your question is yes. We found actually some metastases, but it were so few that it was statistically very insignificant. So we could not do one screening for so few metastases that we found.
Cool. Thank you so much. Brilliant.
Thank you.
Ross, do you mind unmuting yourself, please, to ask your question?
Sure. Chia, that was fabulous.
Hi, Ross.
How you doing?
Nice to see you.
I, of course, love the direction you're going with that. So my question is, so Notch is, excuse me, also losing my voice. Notch is associated with transformation in TALL, as you say. In fact, I think it was discovered, that truncation was discovered in TALL. But in epithelia, Notch has been argued, Notch1 mutations have been argued to actually be protective. You've probably seen this, and we all walk around with Notch mutations in our skin and so on, and don't seem to get cancer. So my question is, based on that dichotomy, have you looked at other tumor types with some of the drug hits you've gotten? Because your original screen, I think, was in the eye. I was a little hazy on that. So have you looked at other tumor types? Do you see specificity to things like the hematopoietic system, or are you getting broader rescue?
So the whole screening was done in this Notch AKT paradigm. For those, for the top hit compound, we also check if it works in another model of cancer that we have in the lab, that is IFUL. And IFUL, it's a cancer model that already has overexpression of Notch or overactivation of Notch, but without AKT. Instead, they have mutations in PSQUIC and LOLA, which are epigenetic regulators. So the top hit compound that we found in the screening, it's not actually working in this other fly model that has Notch activation, but doesn't have AKT activation. So probably this, together with the rest of the results, indicates that this compound that is working so well, it's doing it through AKT pathway, or inhibiting lipoxygenase and inhibiting nitric oxide that are downstream AKT and not downstream Notch. I don't know if I answer your question.
You did. You did. That'll work. Thanks.
Thank you.
From Pablo, last one. Are you aware of high throughput screening platforms for multi-behavior in larvae?
Sorry, can you repeat, please?
Are you aware of high throughput screening platforms for multi-behavior in larvae?
Okay. Yeah, actually, I am not an expert in drug screenings in behavior, but I've seen that there are some other drug screenings, for example, to monitorize sleep and locomotor activity, also neurodegeneration, and obesity and feeding behavior. So actually, I put the example of the cancer disease, but you can do drug screenings in whatever biological activity you are interested in. And actually, there are very interesting papers already published that they see they do behavioral assays, and they use this for drug screenings. So it is useful for a lot of purposes, this technique.
Thank you so much, Dr. Lucía. As a biology undergrad, my terminology in Drosophila is as high as yours, as high as the PhD candidates. So thank you so much for your visual representation of your experiments and everything.
Thank you.
There's some more questions, but they'll be available to answer in Slack, in the Slack platform. So please post your questions over there.
Okay. Thank you very much.
Thank you so much. And thank you everyone for participating in this, and the audience, thank you so much for being involved in this, because science is, it's something that everyone has a passion for. And if you continue researching and striving for perfection and, finding out what exactly the human body does with other organisms, I think that's top notch here. So thank you.
Great.
Thank you, Edwin.
Thank you, everyone, for this wonderful first session. Tomorrow we are going to start at 1:30 Ghana time. And as Edwin said, carry on with Slack. You can post your questions there again. If I see any question in the chat that has not been posted, I can copy paste as well. And the conversation in Slack has started, and it can carry on for as long as you want, as well as tomorrow morning European time. And otherwise, we'll see you all at 1:30 tomorrow.
Bye.
Thank you. Bye.
Bye. Bye. Thank you very much.
Bye bye.
Thank you.
Thank you.
Bye. Thank you.
Bye.
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 Bye. Bye. Goodbye. Goodbye.
Bye.
Let me add now. Bye bye.
Nigerians.
Thanks for the inside presentation. Really appreciate.
Bye. Bye. From Ghana to the world. Bye bye.
Are you ready?
Bye. Okay, bye.
Adiós, chicas. Muchas gracias. Mañana nos vemos.
Bye.
Esto es una pasada. Esto hay que hacerlo más veces.
Sergio, muchas gracias a ti, ha estado súper bien. Además, que chulo, ha presentado un montón de cosas, muy bien, muy bien. Me ha gustado mucho, muchas gracias.
Yo haría esto todas las semanas, ¿eh? O sea, esto hay que hacerlo más.
Pues yo no, yo me iría de cerveza todas las semanas.
Una cosa no quita la otra, ¿eh?
Es verdad, es verdad, es verdad.
Hola, yo sí te tengo que llevar de cervezas, te llevo. Y de seminarios ya veremos. Pero de cervezas las que quieras. Isabel, a ti te llevamos una MAU ahí a Inglaterra cuando tú quieras.
Mejor un buen vino.
Oye, de eso en España te podemos llevar cajas y cajas. Bueno, que muchas gracias, que os lo he escrito antes, pero que me encanta que hagáis esto. Esto es una pasada.
Sí, no, la verdad es que genial. Gracias a ti, Sergio, porque son charlas como esas las que les convencen, la verdad.
No, yo encantado. O sea, y a mí lo que más me gusta es la gente con ganas. Los supercientíficos, súper listos, me aburren mucho. De verdad. A mí me gustan los estudiantes y la gente, esta gente que es todo pasión.
Todo
Es una
pasión.
pasada.
Y
La verdad que te digo una cosa, que la charla que ha dado Amos, qué bien, y cómo se ha reflejado donde empezó y donde está ahora. Es
Sí,
que
muy
él
bien.
es el que le hace ver a la gente que, mira, a lo mejor nosotros no tenemos esos microscopios tan sofisticados y no tenemos no sé qué, pero aún así se pueden hacer muchos estudios y ellos están ahora mismo ya en el mundo ahí de la mosca, ¿sabes? Que
Sí, sí.
yo creo que es
No,
una cosa que los otros deben de pensar, joder, ¿por qué no nosotros?
Está cotilleando algún